so much. Let's move to our last theme of the conference, Advancing Biomarker Discovery for TB POC Tests. And we start with Toby Broger from FIND, who will talk to us about the Biomarker Pipeline for active TB detection.
Dear audience, we have a problem. People, companies, donors, investors, and funders invest a large amount of money into TB biomarker research, leading to a number of reports, but very, very little impact. Imagine a recent publication in Nature found roughly 150,000 reports on more than 1,000 claimed biomarkers, but the translation into the clinic was very, very low, with only roughly 100 markers that made it into the clinic across diseases and applications. Often the research is based on poorly characterized samples, and after an initial study or 2 days, very, very limited follow-up and independent confirmation with very little translation into clinically useful tools. With very few exceptions, there is very little knowledge and data sharing, which leads to rediscovery of biomarkers and waste of resources and our time. Another issue is that many studies actually fail to articulate the intended clinical use case and test the biomarker against it. In summary, biomarker discovery and research has very, very limited impact. But what can we do about it? We've started the development of a TB biomarker database that on the one side captures the evidence from the literature, but also links to the fine specimen bank. This allows us to follow up interesting leads that we identified in the literature with well-designed independent validations. And we can hopefully move biomarkers in the pipeline. In interviews with researchers and companies, it became very, very clear that issues or aspects like intellectual property, data privacy and security, data ownership and data sharing are very relevant and need to be addressed. And here I think we can learn a lot from previous efforts like ReSeq TB. Why will this accelerate the translation of biomarkers? By knowing the status of a biomarker in the pipeline, the TB community can focus efforts and resources on the most promising candidates. The availability of raw data sets that we plan to store as well will let us do secondary analysis and hopefully help us to come up with better predictive signatures. Further, we can perform multi-cohort biomarker validation, which has been shown very successfully in the case of gene transcript signatures. In summary, we believe that the TB biomarker database will help us streamlining the verification and validation of biomarker candidates and eventually lead to more useful clinical tools. In a collaboration with McGill, we have started to search for the evidence in the literature. And I'll show some preliminary data in the next few slides. But let's quickly recap what we are actually looking for. We are looking to detect active TB. In the morning, we've heard a lot about latent and active disease progression, but we initially said we want to focus on the detection of active TB because there are still unmet needs there that are very important. Related to active TB, we have 2 WHO-endorsed TBPs. Let's take a quick look at them. TBP1, which is the non-sputum-based detection test, targets a very high specificity so that we can immediately initiate the treatment. In contrast, the TBP2, which is our triage test, accepts a lower specificity but targets a very high sensitivity so that we can rule out disease and refer people to confirmatory testing. It is totally clear that the triage TBP only makes sense if the cost is significantly lower than the confirmatory test. Both tests target should be relatively simple so that they work close to the patient. As said, biomarkers should be always assessed with the end goal in mind, and I find it quite useful actually to keep those end goals in the ROC curve whenever I plot biomarker study. So let's get back to the systematic approach in looking for evidence. We initially searched all target conditions, including risk of progression, detection of active TB, and treatment monitoring, but then focused the full text extraction on the detection of active TB. The goals are to show the status of the TB biomarker pipeline, identify most promising biomarkers and classes for point-of-care test development, and then, last but not least, use the data as an input for the database that we are planning to develop. Included studies include studies that report on diagnostic performance of non-DNA biomarkers with at least 30 participants that were published between 2010 and 2015. In the initial search, we found roughly 3,500 records. By then removing all the target conditions except active TB, we ended up with roughly 1,000 articles that we screened and included then roughly 200, or included exactly 288 studies that described 413 biomarkers or biomarker signatures. Here an overview of what biomarkers were included. It's interesting to see that a lot of studies actually look at host markers with a focus on protein markers. We still see a lot of antibody detection studies, but then also quite a lot of cytokine studies. Protein peaks are mass spec studies. Then we see the transcriptional biomarkers, and then some hematology markers and T-cell activation markers here. There are very few pathogen markers. We do believe they are much harder to detect. And this is the biomarker pipeline. On the very left, we identified 165 studies that only reported statistical significance, like p-values. So very early stage studies. Then we identified 211 biomarkers, not studies, sorry, biomarkers, that were assessed in a case control design. Only 36 biomarkers were assessed in a prospective cohort. And we all know that only one biomarker, which is LAM, was actually reviewed by the WHO in the last 5 years. Based on QUADAS, we also looked at the quality of the evidence and asked very basic questions like, was there a good reference standard, or was the control group appropriate? And where many studies actually have quite a good culture-based reference standard, a lot of studies actually compare healthy controls against TB positive samples, which usually leads to quite promising results, but is not what we are trying to do or should try to do here with regards to the TPP. Let's have a quick closer look of what we have found in blood. Again, our TPP targets. And here are all the studies that we have found in blood. The size of the study is illustrated here. And this just shows that there is a large number of studies, and those were only studies with a good culture-based reference standard. If we remove now the studies with an inappropriate control group, we end up with a more manageable amount of studies